Let us now study about the structure of an antibody molecule. Antibody molecules are actually made up of polypeptide chains. Each antibody molecule has 4 polypeptide chains, 2 heavy chains, these are the 2 heavy chains, and 2 light chains. Hence, they are depicted as H2L2. These polypeptide chains are arranged as a Y-shaped configuration, the letter Y of the English alphabet. It is represented as the Y-shaped configuration. The stem of the Y or the tail of the Y is made up of heavy chains only. Both these are heavy chains, whereas the arms of the Y are made up of both the light chains and the heavy chains. All these chains are attached to each other by disulfide bonds. Then, every polypeptide has a constant part, meaning this region is known as the constant part, whether it is immunoglobulin A, D, E, G or M. No matter which antibody it is, this part is a constant region, whereas this part, that is the tip of the arms, is known as the variable portion, and it is at this variable portion that we have antigen binding sites. If you look carefully at the variable portion, you will see that the variable portion has the N-terminal and the remaining end has the C-terminal. So this is a structure of an antibody molecule. I'll just repeat it once again. Each antibody molecule has 4 polypeptide chains, 2 small chains and 2 heavy chains. Hence, they are represented as H2L2. Chains are arranged as a Y-shaped configuration. Stem of the Y is made up of heavy chains, while the arms are made up of both the heavy and the light chains. Chains are attached by disulfide bonds. The antibody has a variable portion in the arm called as a V region, and the terminal part of the V has the N-terminal, which has the antigen binding site. It is using this antigen binding site that the antibody binds itself with the antigen, thus neutralizing the antigen. The remaining portion is called as the C portion. Let us now study about the immune system. We had earlier discussed that just like other systems in our body, we also have an immune system. The immune system comprises of lymphoid organs, tissues, cells, and antibodies. We learn more about lymphoid organs. Tissues, I am sure you remember that the connective tissues have macrophages, which play a very important role in our immune system. Cells, the T cells and the B cells play a very important role, as well as the antibodies. We have already discussed the structure of the antibody. Let us now discuss what is a lymphoid organ. Organs where T cells and B cells originate, they mature and they proliferate are called as the lymphoid organs. So they have to form, they have to mature and they have to proliferate. All such organs are called as the lymphoid organs. Lymphoid organs are categorized into primary lymphoid organ and secondary lymphoid organ. Let us see what is the difference between the two. Primary lymphoid organs are those organs where immature lymphocytes differentiate into antigen-sensitive lymphocytes like the T cells and the B cells. You must remember that the immature lymphocytes were formed in the bone marrow, and then some of them migrated either to the thymus and some of them remained in the bone marrows. So, those organs into which these immature lymphocytes migrate and they differentiate are called as the primary lymphoid organ. So, we have two such examples. One is the bone marrow. All the blood cells, including the lymphocytes, are produced in the bone marrow, and the B cells are differentiating themselves in the bone marrow. So, bone marrow is an example for a primary lymphoid organ. Which other organ does such a similar function happen? The thymus. The thymus, as we learned earlier, is located below the heart, beneath the sternum, and we found a group of lymphocytes which have developed in the bone marrow, migrating to the thymus and differentiating themselves into T cells. So, the thymus is also an example for a primary lymphoid organ. Let us now discuss what is a secondary lymphoid organ. Secondary lymphoid organs provides sites for the interaction of lymphocytes, meaning the T cells and the B cells, with the antigen, where they proliferate to become effector cells. Or in other words, where the T and the B cells migrate and interact with antigens and proliferate to help us in bringing about immunoresponse. So there's a difference between a primary and a secondary lymphoid organ. These are actually places where the T cells and B cells migrate and interact with the antigen. Let us see what are the examples for a secondary lymphoid organ. Spleen is an example. Spleen is an organ which is located very close to the stomach, and it contains a lot of lymphocytes, phagocytes. It is also capable of filtering blood-borne microbes. If there are microbes in the blood, the spleen acts as a very effective filter. Other than that, the spleen also is a reservoir and a graveyard of RBCs. RBCs live for 120 days and finally it reaches the spleen, where they are finally destroyed, degenerate. They degenerate within the spleen. So spleen is an example for a secondary lymphoid organ. Lymph nodes are also examples for secondary lymphoid organs. These lymph nodes are found as solid structures all along your lymphatic system, and they are also capable of filtering blood, trapping microbes, and producing primary immunoresponse. So this is a secondary lymphoid organ. The third example is the tonsils. Tonsils is also a secondary lymphoid organ. The fourth example is Peyer's patches of the small intestine. The small intestine has patches called as the Peyer's patches, and all these are typical examples of a secondary lymphoid organ which provides sites for antigen to interact with them. The next example is appendix. Appendix, which you know, is found at the junction between the small intestine and the large intestine. And the last one is the MALT. The full form is mucosal associated lymphoid tissue. Now please understand that 50% of the lymph system, the lymphoid system in our body is made up of MALT, and it is seen in the digestive system, respiratory system, as well as the urinogenital tract, and this consists of almost 50% of lymphoid tissues in our body. So MALT is also a very important secondary lymphoid organ. So this is the difference between a primary lymphoid organ and a secondary lymphoid organ. Let us now discuss about different types of immunity. We have already studied about inborn immunity as well as the innate immunity as well as the acquired immunity. Other than those two types of immunity, our body also has two other types of immunity which we develop. One is known as the active immunity and the other is the passive immunity. Let us see what is the difference between active and passive. An active immunity is when we are exposed to a living or a dead microbe or any other protein. As I told you, it could be a microbial, a molecule or a determinant. If we are exposed to a living or a dead microbe, our body immediately is made aware of that particular entry of an antigen, and our body starts producing antibodies against that antigen. This type of immunity which develops in our body actively is known as the active immunity. Our body is actively involved in production of this immunity. Now this is actually a very slow process and it takes time for becoming fully effective. So the antibodies are produced by our own body, but it is a very slow process. And some of the examples are, if our body is deliberately injected either with a weak or attenuated microbe during vaccination. Remember, when all of us were small children, we have been deliberately injected with some weak microbes. And due to this weak microbe that is injected into our body immediately recognizes that microbe as a non-self substance or as an antigen. And against this antigen, over a period of time, antibodies are actively produced. But it takes time. It is a very slow process, but then we actively start producing antibodies against this deliberate introduction of a microbe. Another example is, suppose we are exposed to an infectious microbe, for example, a chickenpox microbe. Then immediately our body starts producing antibodies against the chickenpox virus. Here again it is our antibody it is our body which is producing the antibodies. So this type of immunity in which we ourselves develop antibodies is known as active immunity. Let us now discuss about what is passive immunity. As the word suggests, here our body does not have an active role in the production of antibodies. We are only receiving ready-made antibodies which is produced by some other organism. We are just receiving it. We are merely recipients of the antibodies. Our body does not produce antibodies but merely receive the same. For example, and this is a very fast process. Unlike the active one, it is a very fast process. Antibodies are produced by some other organism. For example, I'll give you examples like when all of us were very young, the first formed milk that is given by our mother is known as colostrum, and this colostrum has a lot of immunoglobulin A, IgA, antibody A, is passively received by each one of us from our mothers. So that is one way of developing passive immunity. Another method is, when we were all small fetuses, we have received IgG, antibody G, from our mother through the placenta. So both these are passively received antibodies which all of us have. So both are different. One is we are developing antibodies by our immune system, whereas in passive immunity we are just receiving it from some other source. Let us now discuss about the different methods of immunization or vaccination. The principle of immunization or vaccination is based on the property of memory of our immune system. I hope all of you remember what is memory. It is one of the 4 concepts that we have learned as far as the immune system is concerned. Immunization or vaccination is again of 2 types: the active immunization and the passive immunization. Now, what exactly is active immunization? When our body is deliberately receiving inactivated or weakened or attenuated pathogens. Because our body receives these antigens, our body immediately starts producing antibodies against that particular antigen. And when we are actually exposed to a very virulent form of this particular pathogen, then the generated T cells and B cells recognize this pathogen quickly, and on subsequent exposures immediately will produce large scale production of antibodies which can protect us in later years of our life. A typical example of this is measles. Children, when they are very small, are given the injection for measles, the vaccination for measles. This is actually a deliberate introduction of an inactivated or weakened measles viruses. You may have seen that when small children receive this injection, probably that day in the evening or next day, they develop a mild fever. Now, this is actually the primary immunoresponse. So, your body's immune system is actually recognizing the measles antigen, and in turn it is developing a large amount of T cells and B cells. Now these T cells and B cells remain in the body. Suppose this particular child happens to come in contact with a virulent form of measles virus at a later stage in his or her life. Immediately what happens is the massive production of antibodies by the T cells and B cells can immediately kill the antigens and protect the child through this particular kind of immunization or vaccination. So this is one type of vaccination. Another type of vaccination is deliberate introduction of dead pathogens. As in the case of polio, the pathogens which are introduced into a child's body are dead pathogens. But though they are dead, the antigenic determinants or the molecular structure of the polio is recognized by the immune system, and it triggers up the production of a large amount of antibodies which are stored. And subsequent exposure to the same kind of virus at a later stage in life will definitely help in protecting the child against this deadly disease polio. So both these you will find are active kind of immunization, where the body is actively producing antibodies to help the person at a later stage in life. Now, how is it different from passive immunization? In passive immunization, when a person is infected with some deadly microbes to which the quick immunoresponse is required, we cannot wait. We cannot wait because the pathogen happens to be very, very deadly. In such a situation, the person will receive preformed antibodies. We have already discussed that there may be some other organism. Usually in earlier times, the deadly pathogen was introduced either to a sheep or a chick, and the sheep or chick in turn will start producing antibodies for that particular pathogen. These antibodies are then collected, it is purified, and finally the person receives antibodies. So this kind of immunization is known as passive immunization. Typical example is antitoxin for snake venom. When a snake bites a person, we cannot wait for the body to produce antibodies. We have to quickly act so that the patient is saved. Similarly, tetanus. Tetanus is also a very deadly pathogen. So here again, the patient receives preformed antibodies which is produced by some other organism. Salmonella food poisoning is also another example of passive immunization. So, passive immunization is not given to everyone. Unlike active immunization, we have an immunization schedule like DPT, OPV, MMR, etc., which is protecting the child later on. But in passive immunization, the person receives the antibodies only when it is required. Now, nowadays we find that we are using genetically modified organisms. GMO is genetically modified organisms. GMO like yeast. Yeast is utilized, and this yeast is, it is modified with the hepatitis virus. It is modified so that the yeast, through the rDNA technique, by the recombinant DNA technology, starts producing vaccine against hepatitis. So, large scale production for hepatitis B vaccine is now possible through the yeast. Now, genetically modified organisms are used not only, it is not only yeast, but even some other bacteria are used for production of vaccine in a large scale. But this is also passive immunization because some other organism is producing the antibody and we are just receiving these antibodies. We pass on to another topic, autoimmune diseases or autoimmunity. We have learned earlier that our body recognizes self but fails to recognize non-self. Now that is a basic concept. However, in certain instances, maybe it is due to genetic reasons, or maybe it is the body's inability to recognize our own self cells. In such situations, when the immune system fails to recognize self from non-self, what happens is the self cells are destroyed. The body's immune system starts attacking our own self cells. Depending upon which self cells are attacked, there are different types of diseases which result, which are collectively called as autoimmune diseases. The immune system is attacking our own body. So, such a group of diseases are called as autoimmune diseases. Let us see what are they. Suppose your immune system fails to recognize the synovial fluid between the joints. You have learned that our movable joints contain the synovial fluid. Suppose the synovial fluid is considered as non-self. The synovial fluid is gradually attacked and there is a reduction in this fluid, leading to a disease called as rheumatoid arthritis, and that leads to stiffening of the joints. Another example is sometimes the immune system fails to recognize our muscles and it attacks the muscles. Depending upon which muscle is attacked, there are a group of diseases. One common kind of disease is myasthenia gravis. The facial muscles are affected, some other muscles are affected, and the disease due to the degeneration of muscles is known as myasthenia gravis. Let us discuss the next disease. Sometimes our body fails to recognize our own thyroid cells, and that causes degeneration of the thyroid cells. The resulting disease is known as Hashimoto disease. When the myelin sheath in our body is not recognized, what happens is it leads to multiple sclerosis. I am sure you would remember myelin sheath are certain sheaths, insulating sheaths found in our neurons. And when these neurons, the myelin sheath of the neurons are destroyed, it leads to multiple sclerosis. Sometimes our immune system fails to recognize our own liver cells and it starts destroying our liver cells. And that leads to a disease called as chronic hepatitis. So these are some examples for autoimmunity or autoimmune diseases where our body fails to recognize or our immune system fails to recognize our own self. Let us now discuss about allergy. Sometimes when we visit a particular place which is covered with dust, suddenly many of us start sneezing. Or sometimes suppose we are exposed to say hay, we develop rashes all over the body, it leads to itching. Now all these are called as allergy. Now what exactly is allergy? Allergy is an exaggerated response to a hypersensitive antigen. Your immune system is responding in an exaggerated manner. And to what is it responding? It is responding to an antigen for which you are hypersensitive. So the exaggerated response to a hypersensitive antigen is known as allergy. Now, what is a hypersensitive antigen? Antigens which evoke this kind of response are known as collectively called as allergen. So, there are different types of allergens and they can range from pollen grain to dust to animal dander. What is animal dander? Animal dander is the fur and the dead skin which is released from animals like the dogs and the cats. It releases animal dander. Hay or the straw. Food products, some of us have hypersensitive reactions to certain kind of food products like peanuts, shellfishes, egg white, etc. See, prawns, shrimps, etc. are highly allergic to certain group of people. Now, please understand that everyone need not be allergic to a particular substance. Allergens differ from person to person, and the hypersensitive reaction also differs from person to person. Now let us see what exactly happens. When an antigen, any of these antigens, enter into a person's body, immediately it starts being an antigen, it starts producing antibodies. And the antibody which is produced in the case of allergy is IgE. Now this IgE which is produced combines with the antigen antibody combines together to form an antigen antibody complex, and this in turn goes and attaches itself to the mast cells, and a small primary immunoresponse is experienced by the body. So this initial exposure and the combination of the antigen antibody complex and attachment of it to the mast cell is known as the sensitizing dose. So the body is first sensitized. Next time or the second or subsequent exposure to the same antigen will cause the antigen antibody bound mast cell to burst, and certain chemicals like histamines and serotonins are released from the mast cells. So this happens only in the second exposure. Now what happens? When it is released, when chemicals like histamines and serotonins are released, certain allergic reactions are set in. And what are the typical symptoms or the typical um, allergic reactions that result are watering of the eye. The eye starts watering. Then there is sneezing. These are some common allergic reactions: sneezing, running nose, itching of the skin, as well as development of certain rashes, as well as difficulty in breathing. Sometimes the reaction becomes very, very acute and the patient develops severe breathing difficulties. So these are all referred as allergic reactions. Now, what should be done when there is a case of allergy? There are drugs for controlling allergy. Antihistamine, since histamine causes the allergic reaction, a group of drugs called as antihistamines can be utilized. Similarly, adrenal injections and steroid injections can also protect the patient from developing complications, because sometimes when there is difficulty in breathing, it can lead to complications. So antihistamines, steroids and adrenaline can quickly reduce the symptoms of allergy. Nowadays it is seen that the modern lifestyle, as well as since children are living in metro cities, there is more chances of allergy seen in small children, probably because of the sensitivity, probably because of the high pollution rate that is seen in metro cities. There is a more chance for allergic reactions seen in small children. Now, how do we test whether a person is allergic or not? There are a group of tests called as allergic test. Now, what exactly is this test? The patient is either exposed or injected with small doses of possible allergen, and the reactions are studied. Normally, the forearm or the upper arm is, when a person goes for these allergic tests, either in the forearm or the upper arm, small doses of allergen are introduced, very small doses, and it is immediately coded. That particular spot is coded, and then later on, after say 2 to 3 hours or 1 to 2 hours, that particular site is studied to see if there is any kind of inflammatory reaction or any other allergic reaction. And if there are any kind of reactions that are seen in the particular region, the patient is advised to avoid that particular allergen in future, because sometimes that particular allergen may lead to severe complications. So this is the test for allergy.